DNA replication, that is the multiplication or duplication of what? DNA. That is, one double-stranded DNA becomes two double-stranded DNAs. And when we observe this newly formed DNA, that is the daughter DNA, only one strand is the new strand and the other strand will be the parental strand itself. Okay, and that is why the DNA replication is called as semi-conservative. That is, it is conserving the parental strand. And then this occurs during S phase of, that is the synthetic phase of cell division. And then its duration, E. coli, it is about 38 minutes. That is 2000 base pairs are added per second. While in humans, it is about 5 to 6 hours. That is 50 base pairs are added per second. Okay. And then the requirements for DNA replication. The first requirement is the template DNA. Template DNA means the DNA from which the new strand is forming. The new DNA is forming. This is the template DNA. And then the next requirement is RNA primer. This RNA primer is a short stretch of RNA before DNA replication. Okay, that is, before the replication of DNA, a very short stretch of RNA is needed, and this is called as RNA primer. That is, the RNA primer is needed for the initiation of DNA replication. The next requirement is deoxyribonucleoside triphosphate. This acts both as the substrate as well as the energy provider. That is, the DNA replication is energy required process, and this energy needed for DNA replication is given by these molecules. That is deoxyribonucleoside triphosphate, which is of different types. That is deoxyadenosine triphosphate, DATP, then deoxyguanosine triphosphate, that is DGTP, then DCTP, and then DTTP. Different kinds of enzymes are needed. The first enzyme required is helicase. So from the word itself, we can connect the term. This is helix breaking enzyme. We know the DNA strands, they are helically wounded. That is, they are connected by the hydrogen bonds. So for the separation of the DNA strands, the first requirement is the breaking of this hydrogen bond. And this is done by the helicase. That is the helix breaking enzyme. The next enzyme involved is topoisomerase, which is in eukaryotes, or gyrase, which is present in prokaryotes. Okay, this is involved in reducing the tension of the helix. And since the two strands are helically wounded, there is some kind of tension, and this tension will be reduced by the action of topoisomerase or gyrases. And that is why these enzymes are called as superhelix relaxing enzymes. And then another type of enzyme is involved, that is SSBP, a single strand binding proteins. And this is the enzyme that keep the strands apart, or it prevents the rejoining of the DNA strands. Then RNA primase, and this is involved in the production of RNA primer. And then DNA polymerase, which is involved in the replication of DNA. That is the polymerization of DNA. And then another thing is, this RNA primase is called as DNA dependent RNA polymerase, because it is making RNA. That is why it is called as RNA polymerase. But it is DNA dependent. That is, it is making RNA from DNA. Since DNA polymerase is making new DNA from the DNA strand, it is called as DNA dependent DNA polymerase. Clear? That is, the RNA primase is called as DNA dependent RNA polymerase, while the DNA polymerase is called as DNA dependent DNA polymerase. And next is the mechanism of DNA replication, or the steps involved in DNA replication. The first thing is the area on DNA, the site on DNA where the replication begins. It is called as oric, that is origin of chromosomal replication. And then the helicase moves over the DNA strand and it finally binds to oric. Okay, and thus the breaking of the hydrogen bonds occurs, resulting the separation of the strands to form a Y-shaped structure called replication fork. And then the action of RNA primase, that is, before DNA replication, a short stretch of RNA is forming with the help of enzyme RNA primase. That is, the next step is the formation of RNA primer. And then the DNA polymerase add nucleotides. One important thing is here, DNA polymerase add nucleotides only in 5-3- direction. That is, the new strand is forming only in 5-3- direction. So the one strand will be a continuous strand. That is, the strand which is along the direction of the helicase will be a continuous strand, and this is called as the leading strand. But the other strand, which is against the direction of helicase, will be discontinuous. And this strand is called as lagging strand, and the fragments of these lagging strands called as Okazaki fragments. And these fragments will later joined by another enzyme called ligase. The leading strand carry one RNA primer, while the lagging strand carry many RNA primers, right? So finally, every RNA primers will be removed and replaced by DNA. And this is with the help of DNA polymerase 1. In eukaryotes, 3 types of DNA polymerases are there: DNA polymerase 1, 2, and 3. And this DNA polymerase 3 is involved in DNA replication, and the DNA polymerase 1 is involved in the replacing the RNA primer with DNA, and then DNA polymerase 2 is involved in repair of DNA. Then you have to know the polarity of the parental strand. That is, the polarity of the parental strand that makes the leading strand will be 3 dash 5 dash, and the polarity of the parental strand which make the lagging strand will be 5 dash 3 dash. Okay, this is about DNA replication.